In our last lecture, I introduced the concept of homeostasis, which refers to an organism's ability to maintain a constant internal environment within a relatively narrow range of physical and physiological conditions, even though its external environment might be varying considerably. Now, remember from that lecture that in order for an organism to do this, it needs three basic components. First, it needs some sort of receptor, a receptor that will detect the current state of the parameter that the organism is trying to regulate. Second, it needs one or more effectors. These are things that can change the state of that parameter. And finally, it needs some sort of regulator, or what we call a comparator. This is a mechanism that determines the difference between the current value of the parameter and the desired set point for that parameter, and then turns on or off whatever effector causes the parameter to return towards its set point. Now, I ended the last lecture by noting that we also need some way to transmit information between these components. The receptor needs to send information to the comparator, and the comparator needs to send information to the effector. What's the nature of these physiological signals? What is this physiological information? That is, how do these different components tell each other anything that they need to know? Now, earlier in this section of the course, we talked about how cells can detect and respond to molecular signals in the context of our discussion of cell signaling. And in the last few lectures, we've also talked about the role of such signals in development. But we haven't yet addressed in general the problem of how information gets sent from one part of an organism to another part, from one tissue to another, or from one organ system to another. Indeed, the ability to transmit physiological information from one tissue or organ to another, sometimes over very long distances, is essential for the very existence of complex multicellular organisms. And how this physiological signaling works raises an important set of questions that we need to address if we're to appreciate fully how organisms function. And it's this set of questions that we're going to begin to address today and continue with over the next couple of lectures. Now, although mechanisms for long-distance internal communication have evolved in all multicellular organisms, they're really most well developed in animals, as compared to, for example, plants or fungi. The simple reason for this is because animals tend to move about. Now, why should a tendency to be able to move have led to the evolution of more complex internal communication systems? Well, actually, there are a couple of answers to this. One is that because animals move, their environment can change very rapidly. And because the environment can change rapidly, animals need to be able to obtain and process and transmit information about the environment in order to respond in an appropriate adaptive fashion. Also, the variability of animals to move depends on their ability to transmit information, as we'll see later in this section of this course. So, for this reason, we're going to focus our present discussion of physiological signaling on animal signals. We'll come back to plants a little bit later on, but right now, let's just talk about animals. Two structurally and functionally overlapping systems of internal communication have evolved in animals. These are the nervous system on the one hand and the endocrine system on the other. Now, the nervous system is sort of a high-speed, high-performance information transmission and processing system. It involves cells with specializations for the transmission of electrical signals, and these signals travel very quickly. And these cells also have specializations for transmitting an electrical signal from one to another cell. The nervous system of an animal is most intimately involved with the immediate control of what we would call the animal's behavior. And we're going to return to the nervous system later in the next lecture. Now, the endocrine system is a bit more of a slower information transmission system. The endocrine system involves cells that are specialized to produce chemicals we call hormones. Hormones are compounds that are the compounds that do long-distance internal communication between different tissues of an organism. Now, the endocrine system also is involved in controlling an animal's behavior, but it tends to have more global, long-term effects on behavior, and those effects are generally felt at a slower rate. Hormones also play a key role in homeostatic regulation in many physiological systems, as well as control of developmental processes. Now, whereas nervous systems, strictly speaking, are found solely in animals, both plants and animals use hormones for internal communication, especially in development. Well, we've already looked at how cells can signal to each other on the molecular level when we talked about cell signaling a bit earlier in this section of the course. But let's now put chemical signaling into a broader physiological context. The endocrine system of an animal refers to specifically those tissues and organs associated with chemical signaling on the scale of the whole organism, as opposed to chemical signaling between just adjacent cells. Cells involved with endocrine function are specialized to produce chemicals that have a relatively narrowly defined range of signal functions. And these are the chemicals that we call hormones. Endocrine cells are usually assembled into organs that we call endocrine glands. Now, one characteristic feature of endocrine glands is that they typically include some mechanism for releasing the hormones they produce directly into the circulatory system, that is, directly into the bloodstream of the organism. In this way, chemicals that are released by endocrine cells are delivered to the rest of the organism's body very efficiently. In other words, hormones, once produced, go everywhere. Because they're released into the bloodstream, chemicals produced by endocrine glands can exert their action over very long distances, reaching targets throughout the body. Now, I should point out here that not all chemical signals that are specialized for regulation and control of physiological systems necessarily exert their influence over very long distances. And it's instructive to consider a couple of other examples here as a contrast to the long-distance signaling that we'll talk about in terms of endocrine function. For example, so-called paracrine signals. In contrast to endocrine signals, signals that are affecting cells more adjacent to the source. These are signals that behave much like hormones in the way they control physiological response, but only in the immediate neighborhood of the cells that produce them. Another term for these signals is a local regulator. And now, an example of a local regulator is the compound histamine. This compound is responsible for causing inflammation, a tissue response that normally helps protect an animal's body from invasion by foreign materials or microorganisms. Histamine is released in tissues that have been damaged from specialized cells called mast cells. Mast cells store histamine in large amounts in vesicles and release this chemical when the mast cell detects an injury or a toxic substance. When a damaged tissue is inflamed, it becomes, as you're probably aware, red and hot and swollen. Histamine causes this physiological response by causing small blood vessels in that tissue, so-called capillaries, to dilate, to expand, and in fact to cause the walls of those capillaries to become leaky. Literally, the components of the blood leak out through the walls of the capillary because of the action of histamine. This leaking allows protective blood proteins in the blood and white blood cells as well to leave the capillaries more easily and to begin to repair the injured area. Now, although local responses to histamine are protective, we're actually all aware that sometimes histamine responses can spread over larger areas of the body, causing some problems. For example, if you happen to suffer from hay fever, this is because the cells of your respiratory passages are particularly sensitive to some type of pollen. When they have that pollen invade them, they release an enormous amount of histamine and become inflamed. Your nose becomes red and swollen and leaky. And how do you relieve the symptoms of hay fever? You take drugs that counteract histamine, drugs we call antihistamines. Now, actually, we've already talked about another general kind of local regulator a couple of lectures ago when we were talking about development. The diffusible chemical signals that affect the developmental fates of tissues, for example, signals produced by the apical ectodermal ridge and the zone of polarizing activity that we discussed in the context of the development of chick wings, also are kinds of local regulators. These are kinds of local regulators that essentially influence the growth or the fate of a cell in a developing system. Now, yet A very different kind of chemical signaling is that signaling that's specialized for close range interaction between cells in the nervous system, a signaling we call synaptic signaling. This involves the release of particular chemical signals that we call neurotransmitters that come from one nerve cell and influence the activity of an adjacent nerve cell. Essentially, neurotransmitters are the chemicals that allow nerve cells to talk to each other, to transmit an electrical signal from one cell to another, but to do so by converting that electrical signal first to a chemical signal that passes between the cells. And we're going to return to that mechanism in more detail in a couple of lectures. Now, one last general point that I want to make about chemical signaling is that although I've talked about hormones and local regulators and neurotransmitters as separate classes of chemical signaling mechanisms, the lines separating these categories really aren't very distinct. For example, some particular chemicals function both as a hormone in one context, that is, a chemical that exerts its action over a very long distance, and as a neurotransmitter in a different context, a chemical that only exerts its action over the very, very narrow space between two neurons. And most of the molecular mechanisms that underlie the production and action of all of these chemical signals, that is, how cells produce them, how they release them, and how they respond to these signals, are shared in common among all types of chemical signaling. Well, the number of specific different kinds of hormones that you find in a typical animal is really enormous. You can go through lists and lists in textbooks of different specific hormones that have very specific actions on different tissues. And it would be impossible to do justice to any kind of comprehensive overview of hormones and their function in the short time we have available. So I won't attempt that. But instead, what I want to do is to talk about hormones more broadly, and we can do this by classifying hormones into two major groups based on the mechanism by which they interact with the target cells they influence. One broad group of hormones are referred to as the water-soluble hormones. This group includes hormones that are peptides. Peptides are very short proteins, in other words, very short strings of amino acids. And also this group of water-soluble hormones includes a number of compounds that are essentially chemical derivatives of particular kinds of amino acids, or even particular single amino acids themselves. This group of hormones is therefore sometimes also called the peptide hormones. These hormones are called water-soluble because they're generally charged, electrically charged, therefore they are soluble in water. But as we'll see in a moment, because of that, they can't cross cell membranes. Now, the other broad group or class of hormones are called the lipid-soluble hormones. Hormones in this group primarily are kinds of what we call steroids, steroids which are themselves a kind of lipid molecule. Now, the most notable steroid hormones, ones which we're all familiar with, are the so-called sex steroids, which includes compounds like the androgens and estrogens, which are involved in the expression of secondary sex characteristics in development, in the regulation of reproductive physiology, and in the control of sexual behavior. Now, let's talk about how these kinds of hormones interact with cells. And as you'll see, some of this is going to be a bit of a review. Water-soluble hormones, because they're large electrically charged molecules, as I said, can't cross the cell's membrane. And so the way that they have to interact with a cell, the way that they have to transmit their signal to the cell, is by binding to a receptor on the outside of the target cell's surface. Thus, water-soluble hormones interact with cells through the same kind of receptor and signal cascade mechanisms that we already discussed earlier in the context of cell signaling. To briefly review that, the hormone binds to a transmembrane receptor, that is, a protein that spans through the entire membrane, and that transmembrane receptor, once it binds the hormone, transduces what is an extracellular signal, the hormone, into an intracellular signal. The intracellular signal, which is usually a cascade of interacting proteins or other molecules, includes things like the G proteins that we talked about earlier, or second messengers like cyclic AMP. These G proteins or second messengers will in general go on to modify the cell's activity through a signal transduction cascade. Now, I don't want to go into the details of these molecular mechanisms again because we've already covered them, but there is one point that's worth making here. Remember from our discussion of cell signaling that an advantage of having an internal or intracellular signal transduction cascade, such as a cascade of a number of different protein kinases, as we discussed, is that an initially small external signal or very weak external signal can become greatly amplified inside the cell. Hormones are generally released into the body in relatively small quantities. And then because they spread throughout the body, through the circulatory system, they end up having very, very low concentrations, even in those tissues which are their targets of action. Intracellular signal transduction cascades can amplify these vanishingly small or weak hormone signals. To repeat an example I gave earlier when we were talking about cell signaling, a single molecule of the hormone epinephrine reaching a liver cell will cause on the order of about 10 to the 8 molecules of glycogen phosphorylase to be activated inside the cell. That's the end point of that particular signal transduction cascade. Now, the point here is not to remember the details of that mechanism, but to realize that the ratio between the original signal, one molecule, and the end point is a ratio of 1 to 100 million, a huge amplification. Now, in contrast to peptides or other water-soluble molecules, lipid-soluble hormones such as steroids are able to pass through a cell's membrane. And this is because these lipid-soluble hormones are themselves a kind of lipid. So it's possible for these compounds to just simply move through the membrane without any special mechanism to help them exert their influence. Now, because these lipid-soluble hormones can cross through the cell membrane, there has to be some other mechanism to account for the observation that, like the protein or water-soluble hormones, steroids only affect particular target cells. In other words, how could steroids have a specific action on only a certain number of tissues or cell types if, in fact, they get into all of the cells in the body, which indeed they do. They just diffuse across cell membranes wherever they go. Again, this specificity comes from the fact that only the target cells of those hormones have an appropriate receptor, but in this case, the receptor is a receptor on the inside of the cell, a molecule located in the cytoplasm of the cell itself. Thus, when the steroid moves into the cell, it will find and bind with this receptor, if it's present, forming what we would call a hormone receptor complex. And it's the hormone receptor complex which then exerts its action on the cell. Now, how do these steroid hormone receptor complexes affect the behavior of the cell? Well, it turns out that in general, what they do is move directly into the nucleus and activate the DNA. And they do that because the hormone receptor complex acts as a transcription factor. And in this sense, then, these lipid-soluble hormones are doing away with the middleman of the signal transduction cascades that are the way that water-soluble hormones work. Okay, well, now that we've gotten an overview of chemical signaling in general, let's look a little bit more closely at how hormones function on the organismal level. That is, let's look a little bit more at the physiology of hormonal signaling. Now, we defined a hormone as a chemical message that's secreted by specialized cells and distributed throughout the body by the circulatory system. And this raises really what's the first important feature of the way that hormones exert their action, one that we've already alluded to. The hormone, once secreted, will actually potentially affect or potentially reach all parts of the body, and therefore will come in contact with every cell in the body. Thus, a key feature of the way hormone signaling works is by these specific receptors that cells have, the external transmembrane receptors for the water-soluble hormones, or the intracellular receptors for the lipid-soluble hormones. And only the specific cells that are going to be affected by the hormone, what we call the target cells, have these receptors. This is an important point, even if it's a bit of a review of where we've been before. And it's important because because we need to keep in mind that the specificity of hormone action doesn't involve any exclusive connection between the source of the information and the target of the information or the destination of the information. The way to think about this is to think about hormones as working something like radio or TV broadcast signals. This is TV signals before you had cable. These signals, these broadcast signals, are all around us. In fact, right now in this room, there are radio signals everywhere. Why don't I hear those radio signals? Because I don't have a receptor for those signals right now. If I had a receptor, I'd get the signal. Without it, I don't. And this is exactly the way that hormones work. Now, a second general feature of how hormones work is that different kinds of cells can respond to the same hormone in different ways. That is, some hormones will have a single highly specific function and influence only a single kind of target cell, but other hormones can have a variety of different effects on different kinds of target cells. Again, we've already talked about an example of this when we talked about cell signaling. Remember, I used the action of epinephrine, which is a hormone also commonly referred to as adrenaline, that induces what we called then the fight or flight response. If you're scared suddenly, epinephrine initiates a whole series of physiological responses, including increasing your heart rate, constricting blood vessels, releasing more glucose into your bloodstream, and so forth. All of these coordinated physiological responses essentially preparing you for whatever challenge you've just encountered. This kind of coordinated response is made possible by the fact that different kinds of cells in different tissues may have receptors for the same hormone. In other words, epinephrine receptors are found on cells in the heart, in the gut, in the liver. Those receptors all bind epinephrine, but then depending on the cell, those receptors will trigger a different response, the response appropriate for whatever that cell is supposed to do in this coordinated response. Now, a third general feature of hormone action, which we haven't seen yet, is that many hormones function in so-called antagonistic pairs. In other words, one hormone will have one effect on the system, while a different hormone will have exactly the opposite effect. This kind of antagonism is really characteristic of physiological control systems that are homeostatically regulated. Let me illustrate this point with an example involving the regulation of blood calcium levels. Now, the concentration of calcium in your blood is maintained within a very narrow range of values, much as many other aspects of your blood chemistry. There are two different kinds of hormones that are involved in this regulation. The thyroid gland produces a hormone called calcitonin. Calcitonin acts to reduce the level of calcium in the blood through its action on a number of different effectors. For example, calcitonin causes bone cells to increase the rate at which they deposit calcium within themselves. This hormone also causes cells in the intestine to decrease the rate at which they take in calcium from the foods you eat. Finally, calcitonin also decreases the rate at which the kidney recycles calcium, essentially allowing more calcium to pass into the urine and be eliminated. All of these actions end up essentially reducing the calcium level in your bloodstream. Now, the parathyroid gland, which is actually just a different set of tissues associated with the thyroid gland, produces a hormone called, well, somewhat obviously, parathyroid hormone, or what we would call PTH for short. PTH acts on these target tissues in exactly the opposite way. PTH acts to increase blood calcium levels, for example, by causing bones to increase the rate at which they release stored calcium. PTH causes intestine cells to increase their uptake of calcium from your diet, and PTH causes kidney cells to increase their recycling of calcium, therefore preventing it from being lost in the urine. Note that these hormones both exert their action through the coordinated response of several different targets, similar to what we saw for epinephrine. Note also that both calcitonin and PTH regulate blood calcium level through negative feedback, as we would expect for any homeostatically controlled mechanism. But also note that they do so in a completely antagonistic fashion. Specifically, when calcium rises above some set point, the thyroid detects this deviation and releases calcitonin into the bloodstream, the result of which is to cause calcium levels to drop back down in the blood. If, on the other hand, blood calcium falls below the set point, the parathyroid releases PTH into the bloodstream, causing blood calcium levels to rise again. This kind of antagonistic control offers a couple of clear advantages over having just one hormonal mechanism regulate the response. The first advantage is speed. Releasing an antagonistic hormone, one that has the opposite effect, in order to stop or downregulate a system, is faster than merely waiting for the effects of a first hormone to dissipate, because that hormone is eventually lost. In other words, let's say we only had calcitonin to regulate calcium levels. Well, you could imagine that we could have the thyroid releasing calcium or releasing calcitonin if calcium levels were too high, causing calcium to fall. But what if we have calcitonin being released and now all of a sudden calcium levels have become low for some reason? Instead of waiting for the calcitonin's effects to dissipate, we can release a completely separate hormone which returns it to the set point, PTH, much quicker. The other clear advantage really is a matter of precision. A system that depends on the ratio of one hormone to another can be made to regulate that system much more precisely when the absolute concentrations of the hormones is very low, which is usually the case, as compared to a system that depends on just one regulatory mechanism. Now, a fourth general feature that's characteristic of hormone action is that different hormones may have the same effect on a particular kind of tissue. Let's consider here the regulation of blood sugar levels. When we talked about the action of epinephrine in the context of cell signaling mechanisms, I told you that epinephrine causes blood sugar levels to increase when it's released by stimulating the liver to break down a compound called glycogen. Glycogen is a polymer of glucose, so when glycogen breaks down, glucose is produced by these liver cells, and that glucose is released into the bloodstream, increasing blood sugar levels. It turns out that a chemically unrelated hormone named glucagon, which is produced in the pancreas, has exactly the same physiological effect on the liver as epinephrine, also causing liver cells to break down glycogen and release glucose in the blood. What's happening here is that there are two different types of receptors on the liver cell, one specific for epinephrine and one specific for glucagon, both of which end up triggering intracellular signaling pathways which eventually will regulate the activity, increase the activity of the enzyme glycogen phosphorylase, which is the enzyme that breaks down glycogen. Now you might ask, why does a system need two different hormones to perform the same job? Why not just use epinephrine or glucagon? Well, the reason has to do with the fact that epinephrine and glucagon actually have different functional roles to play in the context of the organism as a whole. Glucagon is involved in long-term homeostatic control of blood sugar levels, actually working antagonistically with the hormone insulin, whereas epinephrine is involved in a short-term emergency response, regulating blood sugar levels. Well, a final point I want to make about endocrine function concerns the evolution of endocrine signaling, and this is just an interesting observation, I think. A remarkable thing about hormones is that the very same chemical signals, and by that I mean molecules with the same or virtually identical molecular structures, are often found to occur in widely divergent species. For example, the hormone thyroxine is found in animals that are as different from one another as clams and frogs and humans, all of which use this chemical as a physiological signal. It appears that many fundamental chemical signals evolved very early on in evolutionary history and then have remained chemically unchanged in spite of the fact that the organisms using these compounds for signals have themselves diversified and diverged greatly over an evolutionary timescale, like the difference between clams and humans. But what's even more interesting about this is the fact that even though the structures of these chemical signals remain the same, their functions have evolved to become very different in these very different animal groups. In the case of thyroxine, for example, this hormone seems to be associated with feeding in invertebrates, although the particular action of it isn't clear yet. It is known to be involved in controlling development in frogs. Specifically, it controls the developmental transition from the tadpole stage to the adult stage in amphibians. And in mammals, such as ourselves, thyroxine helps to regulate cellular metabolism. Exactly the same hormone, three very different functions. It's as though certain kinds of molecules just make good signals, so their chemical structure doesn't change, but they acquire new functions, essentially different kinds of information they convey for the organisms in which they are found.